Hello, ninth standard. Have a nice day. I hope you are prepared well for today's exam. We are continuing revision for organization of tissues. As I said in videos 27, we have learned, we have discussed, we have recalled about the organization of tissue, plants and animal tissues. Now this video number 28, we are going to recall the topic mitosis and meiosis. These are the cell division. I hope you remember this. So now mitotic cell division, there are four stages as the cell which is ready for cell division. It is taking place in the vegetative cell or somatic cells, that is cells other than the reproductive cell. They are taking place mitotic cell division. So the first phase, the cell which is ready for division, it is called interphase. And then there are four stages as prophase, metaphase, anaphase and telophase. Where prophase is the first stage, the cell which is getting ready and then making the chromosomes synthesize further, and that is forming the prophase stage, that is as early prophase and late prophase, and then metaphase, anaphase and telophase. Then the mitotic cell division, as such there are two division. One is karyokinesis, division of nucleus, and cytokinesis, it is division of cytoplasm, that is mitosis. Okay, now what you are seeing this slide, it is the mitotic cell division in a root hair region. As I said, ஒரு சின்ன ஆனியன் இந்த ரூட் டிப்பை தண்ணியில் just soak it for 2-3 hours and then take a thin slice, cut off it and see under the microscope. We find this cells தெரிச்ச மாதிரி இருப்பார், அழகா இருக்கு ஆக்சுவலா. அந்த ஒரு செல்லும் ஒரு டிவிஷன். ஆஸ் யூ நோ, இங்கே பாரு பாயிண்டரால காட்டுறேன், இது ஒன்னொன்னும் ஒரு செல் டிவிஷன். வாட் ஐ எம் ஷோயிங், இது ஒரு டிவிஷன். இந்த மாதிரி ப்ளூல இருக்கிறதெல்லாம் ஒன்னொன்னும் ஒரு செல் டிவிஷன். I hope you understand. So இது வந்து anaphase stage, அது மாதிரி இது ஒரு anaphase stage, இது ஒரு anaphase stage, இது metaphase stage, இந்த மாதிரி different stages. When you see under the microscope, we could see all these different stages of mitosis. சரியா? The next one, it is interphase, where when the cell, before it undergoes mitotic division, it prepares itself for the cell division. This phase is called interphase, where the chromatin material duplicates due to duplication of nucleic acid. Otherwise, in short, the cell which is ready for undergoing mitotic division. Prophase stage, chromatin, they begin to coil and appear as a long thread-like structure. They are called chromosome. Actually, chromosome and it will have two arms as attached with the center centromere. During cell division, it divides into two as two chromatids. So chromatids, they begin to coil and appear as long thread-like structures called chromosomes. Each chromosome consists of two chromatids that lie side by side and are joined along the point called centromere. Then spindle fibers are developed from the poles towards the center and disappearance of nucleolus and nuclear membrane. So the thickening, coiling and forming a thread-like structure, that forms the early prophase, and disappearance of nucleolus and nuclear membrane and appearance of spindle fiber, they form the late prophase stage. Then the second stage is the metaphase, where the nuclear membrane totally disappears and the chromosomes become shorter and thicker. Chromosomes, it becomes shorter and thicker, and the chromatids move to the center of the cell with their centromere, and the centromeres are attached to the spindle fiber. So the metaphase, they are characterized by formation of four chromatids, and they are arranged in the center. Otherwise we say it is arranged on the equatorial plane. And that equatorial plane, when the chromatids are arranged, they are not hanging, they are held by the spindle fibers. That is forming the metaphase stage. Whereas anaphase stage, the centromere of each chromosome divides into two, and with each chromatid gets a centromere, and it becomes a chromosome, and they move to the opposite pole by the contraction and the spindle fibers, they move to the opposite pole. That is anaphase stage. In telophase stage, where the daughter chromosomes reach the pole, and the chromosomes arrange themselves with the reappearance of nucleolus and nuclear membrane, that forms the early telophase. And late telophase, there is a constriction in the cell plate or cell wall appearance, that is called phragmoplast. This appearance, formation of cell plate in the late telophase stage of plant cell, it is called phragmoplast. And then division of cytoplasm, cytokinesis occur, there it forms the two daughter cells. That's the mitotic phase as prophase, metaphase, anaphase and telophase. And cytokinesis, division of cytoplasm, and in plant cell the cytoplasm occurs by the formation of cell plate, and it is called phragmoplast. And cell plate at the center of the cell between the two daughter nuclei. Thus at the end of mitosis, two identical daughter cells are formed. Then significance. This equational division results in the production of diploid daughter cells with equal distribution of genetic material. This equational division results in the production of diploid daughter cells, that is 2n or 2x with equal distribution of genetic material. Then in multicellular organisms, growth, organ development and increase in body size are accomplished through the process of mitosis. Then third one, mitosis helps in repair of damaged and wounded tissues and by the renewal of lost cells. This is the significance of mitosis. I hope you are able to follow this. Okay. Then next we will continue meiosis. Meiosis, again it is a reduction division taking place only in the reproductive cell, that too at the time of reproduction, only during the formation of gametes, as male gamete by the process spermatogenesis taking place in the testis, and female gamete by the process oogenesis taking place in the ovary. As you know, testis is formed of numerous highly coiled seminiferous tubules, where some of the cells, they are forming spermatogonia that undergoes meiosis. Same way in ovary, it is formed of Graafian follicles that on further development and maturation, it undergoes the process oogenesis, producing the ovum. And in female human, the reproduction in female, the production of ova, there are pair of ovaries, but only one at a time. Once left ovary is releasing the ovary this time, next time right ovary. And it will take a turn. Whereas in a testis, there are millions of sperms will be produced at a time. And in ovary, the release of ovum, the process is called ovulation. So here in Meiosis, there are two categories as heterotypic division as meiosis 1 and homotypic division meiosis 2. So this heterotypic division ஏன்னாக்க prophase stage there are 5 different stages as leptotin, zygotin, pachytin, diplotin and diakinesis. I repeat, leptotin, zygotin, pachytin, diplotin and diakinesis. In the 5 substages it is getting the name as heterotypic division. So as prophase 1 with the 5 subdivisions, then metaphase 1, anaphase 1 and telophase 1. Same way meiosis 2 prophase 2, metaphase 2, anaphase 2 and telophase 2. சரியா? So now இதில் prophase 1 leptotin stage the chromosome they are as homologous chromosome. Homologous chromosome na one from father, another from mother. Otherwise we say paternal and maternal. Normally blue and the paternal, red maternal. So they differentiate. And then in zygotin stage they lie side by side. That is called pairing or synapsis. So during this pairing, at point of attachment, at certain places they are attached to each other. So that is in pachytin stage, it is called tetrad. Okay, so that point of attachment, it is called, that point of attachment is called chiasmata. And as a result of point of attachment, there is interchange of segments, thereby exchange of genetic material. Right? So this is pachytin stage where crossing over occurs. Leptotin stage pairing or divalent or synapsis சொல்லுவோம். Chromosome, homologous chromosome like side by side. Pachytin chiasma formation and crossing over. Right? In diplotin stage, as they are pairing, so it is called diplone சொல்லுவோம். And then with the exchanged segment in diakinesis, with the exchanged segments order, the chromosome, they try to, the chromatids will try to move apart. So as they are moving apart, at one stage it terminates. So it is called terminalization. I hope you understand. That stage is taking place. That stage is taking place in diakinesis. Okay. And then metaphase stage, chromosome with the exchanged segment, they are arranged on the equatorial plane. So by the time in diakinesis it gets completed, there is disappearance of nucleolus and nuclear membrane will occur. And then metaphase stage, chromosomes are arranged on the equatorial plane and they are held by the spindle fibers. So அதே தான் diagrammatic representation, leptotin, zygotin, pachytin, diplotin and diakinesis. Leptotin condensation of chromosome, they become clear and thicker. Zygotin description clear இருக்கு, நல்லா படிச்சுக்கோ. Zygotin formation of bivalent, then formation of pairing, synapsis between homologous chromosome, they lie parallel, pairing, synapsis occurs. And in pachytin stage, exchange of chromosome. The chromosome, the blue line the end with the red. அதே மாதிரி இந்த red ending with blue. So this is crossing over. And the point of attachment, it is called chiasmata. Actually இது இங்கேயும் போடலாம். That point of attachment with the exchanged segment, that is called singular chiasma, many chiasmata. Right? And then this separation, that is dissolution of pairing, இது segment, exchanged segment ஆயிடுறது. So then the dissolution of the diakinesis, disappearance of nuclear membrane and nucleolus, that is taking place. That the prophase 1 gets over, and then in the meiosis stage, metaphase stage, chromosomes are arranged at the equatorial plane, and in anaphase 1, by the contraction of the spindle fiber, the chromosome are moving apart and they are moving to the opposite poles. I hope you understand. And then in telophase stage, further reappearance of nucleolus and nuclear membrane. Then the same prophase 1 will undergo second meiosis division, which is similar to mitotic division. Sure, yeah. So the significance, meiosis helps to maintain chromosome number constant. Then in meiosis, 4 haploid daughter cells are formed. Then it forms, it is very important in sexual reproduction. Then it is meant for recombination of genes resulting in genetic variation. And this genetic variation forms the raw material for evolution. That is the cell division, mitosis and meiosis. So you just recollect these points, then revise for further answers. I hope you have understood. So prepare well, write well for the test. We'll continue in the next session. Thank you.